Hi, this is Xinhun Shao from University of Southern California, and I have nothing to declare. Simultaneous acquisition of complementary function responses can reflect different aspects of brain activity. For example, bold fMRI can reflect a complicated combination of underlying CBF, CBV, and CMRO2 changes according to this Davis model. And concurrent measurements of multi-contrast fMRI has already drawn much attention 20 years ago. This study shows bold activation is the strongest with more voxels activated than the vasal and ASL contrast, while vasal and ASL contrast are more focalized compared to bold. Here's another study showing that activation induces a bold response, which is only about 1%, while the underlying blood volume changes is almost 10%, and blood flow increase is almost 40%. And nowadays, with hardware and technical developments, ultra-high spatial resolution fMRI techniques have been developed to study neural activation across cortical layers. For example, in the visual system, there are feed-forward and feedback pathways to process the visual information, which involve different cortical layers to response. We developed a zoomed arterial signal labeling technique and 7 Tesla to measure CBF response and study laminar CBF response to spatial visual attention task. And we found that unattended visual stimuli and visual spatial attention lead to different CBF activation profiles, which corresponds to 2 different pathways respectively. And we further extended our 7T zoom ASL technique for concurrent CBF, CBV, T2 Grace bold, and CMRO2 measurements. And here's the diagram of the pulse sequence. And we use a zoomed acquisition in this smaller volume to shorten the acquisition time. And CBF contrast was generated by the subtraction of ASL control and label images, while ASL labeling plane was placed perpendicular to the MCA, PCA, and AC, those major arteries. And a non-invasive inversion pulse was added to generate visual contrast after a certain pulse-event delay about 1.2 seconds. And the separate Grace bold image was acquired 500 milliseconds after the ASL visual acquisition. Lastly, we will calculate the profile of the CMRO2 response based on the profiles of the CBF, CBV, and Grace bold signal according to those equations. We collected data from 3 subjects on Siemens 7 Tesla Terra scanner, and the visual stimuli present a unilateral half-ring shift pattern with high contrast radio grating that reserved contrast and 4 Hz. Each experiment interleaved 48 seconds of task, followed by 48 seconds of rest. The total acquisition time was 32 minutes. And here shows 5 representative slices of CBF maps acquired in resting state and with task. And you can see very strong activation can be directly observed from those CBF maps. And here are the activation maps of the CBF, of bold and visual. Very strong and similar patterns can be observed between those 3 different contrasts, while the visual shows the opposite response, which is mostly negative in the activation region and positive in the adjacent voxels. And bold response is just very strong, but ASL and visual contrasts are more localized within the gray matter compared to the bold response. And here shows the bold CBF and visual activation maps projected onto the cortical surface, and the ring-shaped bold and CBF activation can be observed corresponding to the pattern of the visual stimuli on the contralateral hemisphere, while decreased bold and CBF signal can be observed in the adjacent regions. And a similar pattern can also be observed in the visual maps with opposite responses. And we manually draw ROIs to calculate the laminar profiles within the negative bold response regions and in the positive bold response regions. And here shows the laminar profiles of the CBF, bold, visual, CBV, and CMRO2 signal changes in the positive response regions, the orange color, and in the negative response, the bold response regions in the blue color. You can see the visual stimuli induced the CBF and bold increase, mostly peak in the middle layers, which is consistent with the previous studies. And the decrease of the CBF and the CMRO2 were mostly predominant in the deep cortical layers. And the amount of the decrease is very comparable to the amount of the increase, which is 71% for the CBF and 75% for the CMRO2, while the CBV decrease, which is 56%, incomparable to the positive increase voxels, while the BOLD decrease is only 7% compared to the increase in the positive BOLD response regions. So in conclusion, we propose a 70 laminar concurrent ASL visual and BOLD fMRI technique to obtain quantitative CBF, CBV, T2 BOLD, and also CMRO2 measurements with high resolution and specificity, and which demonstrate that the technique can be used to study the layer-dependent vascular and metabolic activities. And our study further demonstrated that negative fMRI responses, which were associated with moderate decrease in T2 BOLD and CBV signals, but a very strong CBF and CMRO2 decrease, especially in the deep cortical layers, which could be related to suppressed neural activity. And thanks for your attention.